About 1.6 billion years ago, a larger bacterium sized up a smaller blue-green looking one and engulfed it. However, this time, instead of digesting it and using it for food, it stayed alive within the larger bacterium. What I've explained is the origin of the first chloroplast, or the endosymbiotic theory. A similar process occurred to create what we now know as mitochondria. Over time, the larger bacterium enslaved the smaller microbe by transferring more and more of its DNA into its own genome. However, not all of that DNA was transferred, and to this day, mitochondria and chloroplasts have unique DNA that must be maintained and replicated in order for the cell to survive. So how is this DNA maintained? In our lab, we study several core proteins that are in charge of replicating this DNA. These include Polymerase A, Polymerase B, and Twinkle. Polymerase A and Polymerase B are DNA polymerases that operate in plant organelles. Normally, even the simplest microbe will have many versions of DNA polymerase to replicate its DNA, but plant organelles only get these two. Thus, these two polymerases, Polymerase A and Polymerase B, are in charge of a lot, including DNA replication, DNA repair, and proofreading. Twinkle is a protein with two important functions: unwinding DNA and preparing it for synthesis through a process called priming. If you were to look at Twinkle in three-dimensional space, you would be able to see these two regions distinctly defined. 6 Twinkle proteins will form a ring and unwind DNA by pulling one of the strands through this ring. DNA then passes through the priming region of Twinkle to prepare it for Polymerase A and Polymerase B. Other versions have shown that animal versions of Twinkle, Polymerase A, and Polymerase B interact with each other to replicate organellar DNA. Using several independent methods, we have shown that the same interaction occurs in plant organelles. We have also shown that Polymerase A and Polymerase B interact specifically with the priming region of Twinkle. There is, however, one glaring difference between plant and animal organellar DNA replication. In animals, when Twinkle is mutated, the animal dies. In plants, when Twinkle is mutated, the plants are completely fine. So there are several important questions we need to answer, the biggest being, how are plants fine without Twinkle, such an essential protein? What else interacts with Polymerase A and Polymerase B? What other mechanisms are in place that allows for plants to survive without Twinkle? Tackling these questions will help us distinguish the differences between DNA replication in plant organelles and animal organelles. It will help us understand more about endosymbiosis, the mechanisms in place, and what makes mitochondria in your cells different from mitochondria in plant cells.